I'm Stephen Stacker from the Tumour Angiogenesis program at the Peter MacCallum Cancer Institute in Melbourne, Australia. Our interest is understanding the role of lymphatic vessels in cancer. In normal biology, lymphatic vessels start as a series of blind-ended sacs which progress through pre-collecting lymphatics, finally into larger collecting lymphatic vessels. And these larger lymphatic vessels are surrounded by smooth muscle cells and have active valves. Fluid and cells are then passaged through here to regional lymph nodes. In the case of cancer, lymphatic vessels are remodelled through exposure to a series of protein growth factors. Some of these also cause tumour angiogenesis. So remodelling the vessels occurs at the level of the smaller blind-ended sacs where we see lymphangiogenesis and lymphatic enlargement. In the larger collecting lymphatics, we have observed dilation of these vessels, and that's what I've talked about here at the conference. Tumour cells can therefore leave the primary mass, progress through the lymphatic vessels and then proceed to regional lymph nodes and then potentially further to metastasise into distant organs. It's also interesting to note that changes in remodelling to lymphatic vessels may occur within sentinel lymph nodes as a precursor to tumour cells being transported there. So we've used a mouse model of cancer where members of the VEGF family, the vascular endothelial growth factor family, have been overexpressed. And in these models we demonstrated that when the lymphangiogenic growth factors VEGF-C and VEGF-D are overexpressed and essentially form a metastatic tumour, we see dilation of large collecting vessels between the primary tumour and the regional lymph node. And that's demonstrated here by the uptake of patent blue dye in these vessels. In non-metastatic tumours, so that is either controlled tumours not expressing those factors or those expressing the angiogenic factor VEGF-A, you can see that the lymphatic vessels are not dilated, and that's shown graphically on the right-hand side. So we've used two of these models to try to isolate distinct gene signatures that might relate to the dilation of these vessels and to their propensity to encourage metastatic spread. We've isolated the cells that line these vessels, the so-called collecting lymphatic endothelial cells. We've enriched those cell populations from both metastatic and non-metastatic tumours, and by extracting the messenger RNA and doing a microarray comparison, we've been able to identify candidate genes that might be implicated in this process. One particular gene that we've isolated relates to the breakdown of some of the key prostaglandins within tumours. So prostaglandins are very important molecules for mediating inflammation and vessel dilation. The molecule that we've isolated and studied is called PGDH, or prostaglandin dehydrogenase, and it's a key enzyme in degrading prostaglandins. To cut a long story short, our study has shown an intersection between these key lymphangiogenic growth factors, VEGF-D and VEGF-C, which are overexpressed in tumours and, at least in human tumours, are highly related to metastatic disease. In our models, we've shown that VEGF-D can promote the downregulation of this enzyme PGDH, which causes a lack of degradation of prostaglandin. When these prostaglandins persist in tumours, that causes dilation of collecting lymphatic vessels and encourages metastatic spread. There is also the potential here that simple drugs such as NSAIDs could be used to inhibit this process. Just as a final note, we've also begun studies using high throughput, high contact technology to do genome-wide siRNA screens to examine molecules that might be important for the migration of lymphatic endothelial cells. We've used this model as a surrogate to look at lymphatic vessel remodelling, and we hope by using this type of broad approach we'll identify families of genes that are critical for remodelling of lymphatic vessels, not only in human cancer but in human pathology. So thank you very much for listening.